I want to thank Greg and Maggie for organizing this, and also Boris for inviting me to participate in the Pew Bio summer course. I've spent the past 3 weeks here at KITP leading a group of students through some really exciting cheese-related projects in the lab using some very new technologies that were brought here by other instructors. And so it's just been wonderful to get to spend some time here in Santa Barbara. Let me just tell you a little bit about my background and how I got to where I am. I am a California girl, born and raised here. I grew up in South San Francisco, the industrial city, not really very exciting place, but, and I was a UC undergrad, so not here at Santa Barbara, but in San Diego. So I studied molecular biology at UC San Diego, and then I spent 11 years at Harvard doing my PhD, and then spent, well, not 11 years as a PhD student. 6 years as a PhD student in microbiology, and then I had a 5-year fellowship at Harvard where I was able to, they basically gave me my own research lab for 5 years and said, do whatever you want. And so I said, well, I'd like to study cheese. And they said, great, sounds like a great idea. And it was actually through that program that I initially met Boris, who was on the advisory committee of it. So anyway, so after that 5-year period, I was lucky enough to get a faculty position back at my alma mater, UC San Diego. And for the past 2 years, I've been getting my research lab set up, hiring graduate students and postdoctoral fellows to work with me, undergrads. And so now we have a bustling lab at UC San Diego. And we all just spend all of our time thinking about cheese, which is, not a bad gig. Okay, so I'm going to tell you, I'm going to give you guys a sort of taste of the science of cheese and a taste of the research that we do, and try and combine those in a way that convinces you not just that cheese is even more amazing than you already thought it was, but also that it can be a really useful and interesting model for studying microbes. So I'm going to start with a question, which is why even make cheese? What's the point of making cheese? Because it's delicious. Yes. Anybody else?
Preserve milk.
Preservation, right? Okay, exactly. So cheese making is thought to go back, date back close to 9,000 years. That's our current best archaeological evidence. Originated probably in the Fertile Crescent, around the time of the dawn of agriculture. People had started having herds of animals at that time. And, it turns out that milk is an incredibly nutritious food product. And if you happen to be able to consume milk as an adult, then you've got quite a bit of extra benefit going for you in terms of your nutrition. Now, humans at the time, most humans were not actually able to consume milk into adulthood. Our ability to consume milk as adults only occurred sort of around that same time, but through a mutation in our lactase gene that allowed us to keep producing this enzyme to break down milk. And so one of the benefits that is thought to have factored into the production of cheese is not just preservation, but also the fact that cheese making reduces the lactose content of milk and therefore makes it edible, even if you can't drink milk as an adult. So you can benefit from the nutritional value of milk by making cheese, even if you couldn't drink it otherwise. Okay, so it's incredibly nutritious. Milk is. It has a lot of fat, a lot of protein, a lot of sugar in the form of lactose. And this is great for us as a really reliable source of nutrition, but it's also great for microbes. And so we're often constantly in battle with microbes over food sources in many ways. And if you don't get the right mixture of microbes growing in this milk, as I know I have, and I'm sure many of you have found a spoiled jug of milk in your refrigerator, you know that it can become quite foul and inedible. However, if you know how to do it, you can actually control what types of microbes grow in your milk And instead of spoiling it, you're preserving it and transforming it into this wonderful, delicious food. And so I sort of think of cheese making and other types of food preservation processes as sort of controlled spoilage. So we still want microbes to grow in the food because they're actually doing us a favor by preserving it. But we really need to control what types of microbes do this. And I think it's completely amazing that, 10,000 or even more for probably other types of fermented foods, humans had developed methods for controlling microbiology in a very precise way before we even knew microbes existed, way before we knew microbes existed. And so I sort of think of cheesemakers and other people who've made fermented foods throughout the years as sort of the first microbiologists, the first microbial ecologists. They're farming microbes even though they didn't know they were doing it. And so apart from cheese, pretty much every culture on the planet has come up with ways to harness the power of microbes to make delicious foods that stand the test of time. So whether it's something simple like yogurt, which is in essence very similar to the initial stages of cheese making, pickled vegetables like this delicious pickled cabbage called kimchi, which is originated in Korea, soy sauce, fish sauce, this really interesting and kind of funky product, natto. Has anybody here tried natto? Yeah, so it's fermented soybeans, and it's fermented with this bacterium that produces like a goo, like a slime. And so these soybeans, they have a lot of protein in them, and the bacteria eat the protein and they produce a lot of ammonia. So the beans smell really disgusting and they're slimy. But it's a delicacy in Japan. And has a lot of vitamins that are introduced by the bacteria that are fermenting. Coffee and chocolate. Did you guys know coffee and chocolate were fermented foods? No. Yeah. So thank you, microbes. So in both cases, in chocolate especially, the microbiology is incredibly important for getting the seeds of the fruit to a point where it becomes much more edible and delicious. And the same is true for coffee. So these are both fruits that are fermented first by microbes and then processed by us. Things like pickles, beer and wine, sourdough bread. You can sort of think of all kinds of examples. Pretty much every culture on the planet has figured out ways to do this to our benefit. And so I'll give you guys a quick sort of cheese 101 so you can go home and impress all your friends about the nerdy details of cheese making. Okay, so when we think about cheese making, we have to first think about milk and what milk is made of and what microbes might be doing inside milk to make cheese. So milk, as I said, has a lot of protein, fat, and sugar. The protein, these little squiggly balls, it's mostly casein, so it's this very large protein. The primary reason why milk looks white to us is because there's so much protein in it that's sort of causing the light to not be able to pass through the milk when these casein molecules are in solution. We have a lot of triglycerides in the milk, as well as the sugar lactose, which is a special kind of disaccharide, which many organisms have a hard time breaking apart these two pieces of sugar before they can eat it. So how do you go from this liquid to a solid, basically? So the first step in cheese making is you take this milk, you warm it up, and you add these special kind of bacteria, which I'll tell you more about, lactic acid bacteria. They're called lactic acid bacteria because they actually eat the lactose and produce a byproduct called lactic acid, which sours the milk in a pleasant way. And this enzyme rennet, which I'll tell you a little bit more about. So the lactic acid bacteria, these are often called starter cultures. They're the same things that you see listed on the side of a container of yogurt, so these live active cultures. These are the same types of bacteria that are used in cheese making. And they serve a number of purposes in cheese making. You can see a picture here. These are some lactobacillus and some lactococcus, different bacterial species, and these are all the protein sort of aggregates in the milk. I think this is actually a picture of yogurt. So the lactic acid produced by these bacteria does a number of things. One is that it gives this tart flavor to either yogurt or cheese. So lactic acid is an acid, sort of like citric acid, so you get this sort of tartness. It acts as a preservative because many pathogens can't tolerate this acidic environment. So that's great. And it also contributes to the formation of the cheese curds themselves. So I'll tell you more about that in 1 second. So basically what these bacteria have evolved the ability to do is eat lactose, which I said many organisms can't do this. So I sort of think of these bacteria as lactose-tolerant bacteria. So they produce an enzyme that's very similar to the one that we produce in our digestive tract to break down this disaccharide, lactose, which is two sugars, glucose and galactose, linked together by a bond that's difficult to break unless you have this specific enzyme. They break it apart into these two simple sugars, and then that is inside their cell. They undergo this process of fermentation. So basically they're eating the sugar and producing energy so they can grow, and they spit out as a byproduct lactic acid. Okay, so this is their primary role in cheese making. And then the second important part of this first step of cheese making is the addition of this enzyme called rennet. So rennet is traditionally an extract from the fourth stomach of a milk-fed calf. So, not especially, tasty thought, but mammals have enzymes in our digestive tracts to break down milk because that's the first food that we ever consume. And so calves have this enzyme. The rennet is the extract, the chymosin is the actual enzyme, and this enzyme specifically breaks apart milk protein. And so what happens is in liquid milk, you have casein, which I'm representing as this kind of big protein, and on the surface of casein are a bunch of negative charges. So you can imagine these sort of globules of casein floating around inside liquid milk, and when they encounter each other, they bounce apart from each other, right? Because they're all negatively charged. It's like two negative ends of a magnet coming together and repelling each other. So the first thing that happens is that the acid produced by the lactic acid bacteria starts to neutralize these negative charges. So now the proteins start to come together a little bit. They're not repelling each other anymore. And then this enzyme, chymosin from rennet, actually will go in and chop off the rest of these negative charges from the tails of the protein. So it'll specifically cleave off these outside parts. So the combination of the acid and this enzyme lead to the protein aggregating inside the liquid and separating from the liquid. Okay? So you get this aggregate of casein, and that's basically our fresh cheese. So those aggregates are the curd. So we separate the curd from the whey. Exactly. So the curds we sort of press together to form a fresh cheese. You can do lots of interesting things with those curds depending on what type of cheese you want to make. The whey, the liquid that's drained off, you can also do lots of interesting things with that. Has anybody ever had ricotta cheese? Mm-hmm. Yes. So ricotta traditionally, it means recooked ricotta. You take the whey, there's still protein there, you heat it up, and the leftover protein coagulates into these smaller little curds. So it's a very low yielding product, so you usually use large vats of whey to make a little bit of ricotta. You can also feed the whey to pigs. They love whey. They go crazy over whey. And so if you have large amounts of cheese production going on, you have lots of whey to get rid of. In Parma, Italy, where they make Parmigiano Reggiano, they have lots of whey. So they feed the whey to pigs. The pigs love it. Mm-hmm. It makes especially tender meat, and they use those pigs to make prosciutto di Parma. Mm-hmm. So basically you have this cycle of cheese life. And I think we actually had some prosciutto out here. Yeah. Okay, so you have this fresh cheese, And it's tart, kind of yogurty tart. The curds can be soft or firm depending on how you treat them, but it doesn't taste that different than milk. It's just kind of tart, milky product. So the part where I get really excited is when you take this fresh wheel of cheese and put it into a damp, dark, moldy cave. Which I don't know who ever decided that would be a good idea. Turns out to be like the best idea ever that anybody ever had in the history of mankind. So you take this fresh piece of cheese and you put it in a dark, damp cave, and what happens is that it becomes colonized by a mixture of bacteria and fungi. They grow on the surface of the cheese and completely transform it to all of the stinky, delicious, wonderful cheeses that we love. Okay, so here are just some pictures of some of these amazing cheeses. And one of the fascinating things here is how much diversity we have of cheeses. There's hundreds, if not thousands, of different types of cheese. They represent, to me, each different little ecosystems. And you can see on the surface of these cheeses all these different colors and textures. You can imagine the smells that they have. This is actually one of the cheeses that you guys had tonight, Red Hawk. And so what I'm going to tell you about are some of these amazing microbes that grow on cheese during the aging process. And I'm going to give you a little bit of insider knowledge about how they actually make cheese. Okay, so before I kind of dive into microbes, I'll give you just a quick primer on these mysterious organisms. So this is looking under a light microscope at a scraping from a piece of cheese. And you can see a collection of different types of microorganisms living together. So we have two different types of fungi. So we have molds and yeast. So yeast are single-cell fungi. They're what are used in sort of bread making or beer making or wine making. And then we have molds, which you would often see on an old piece of bread in your kitchen. They form these long filamentous structures. They form aerial structures that have spores on them. Really fascinating organisms. So these tubes that you see are the molds. These are the big structures that they form that kind of can create a mat on the surface of a cheese. And then these larger round circles are the yeast, single-celled fungi. And then we have bacteria, which are these little tiny circles that you see. So they're much smaller than yeast. They're also single-celled organisms. You may think, well, they're all small. They're probably all pretty much the same thing, doing the same thing. However, the fungi, the yeast and the molds, are much more closely related to us than they are to the bacteria. So we are sort of distant cousins of yeast and mold, believe it or not. And bacteria are on a completely different branch of the tree of life. So all small, but very different. And so basically what they're doing as they're growing on cheese, they're just trying to make a living. Okay? They're just trying to eat and grow. That's what they do. And so as I said, we have a lot of protein, fat, and sugar on cheese. Those are all great food sources. One of the things that these microbes can do is they can break the proteins down into smaller and smaller bits. So they're basically chewing on the proteins and eating them. This creates a bunch of interesting flavors. So things like amino acids, which can have very brothy flavors, meaty flavors. The fats get broken down to free fatty acids, which can have sort of spicy, really aromatic properties. And the sugars, I already talked about, can be broken down to acids like lactate. And then each of these byproducts can be further broken down by more microbes to smaller and smaller bits, so small that they actually become volatile, so we can smell them when we pick up a piece of cheese. So all of those things that we smell when we pick up a piece of cheese are basically the byproducts of microbial growth. So we're smelling microbes growing. And see, these are a couple of fun smells that these microbes make on cheese. So these are compounds that have been identified on cheese. This is a compound 2-methylpropanol, which smells like bananas. Methane thiol, which smells like rotting cabbage. Okay. Methional, which smells like roasting meat. Butyric acid, which smells like sweaty gym socks or body odor. Diacetyl, which tastes like butter. And benzaldehyde, which has sort of a bitter almond flavor. And so what I'd like to imagine is going on is that each of these different types of microbes that you find on cheese has their own set of things that they like to eat and things that they produce from what they eat. And so depending on which combination of microbes you have on your cheese, you can have all these different mixtures of flavors and smells. And so that is part of what leads to this incredible diversity of flavors and smells in cheese, is this sort of combinatorial effect of the microbial growth and different food sources. And you may think, well, why would anybody want butyric acid on their cheese? And, some people might not, but, if you are a person who appreciates a stinky cheese, it's because you've sort of learned to appreciate small amounts of the smell in combination with all the other delicious things that are there. So some of these are definitely acquired tastes. Okay, so who are the microbes that are actually doing this? So the first microbe that I'm going to talk about is one of these fungi. So this is a mold. And it's often found on bloomy rind cheeses, just like the one you guys just ate. So notice I'm telling you this after you ate the cheese. So this is a picture of Stepladder Creamery Ragged Point. It's this beautiful bloomy rind cheese. This is actually one of the newest cheesemakers in California. Really wonderful, and they're in Cambria. Cambria? Is that how you say it? Yeah. So local. And so this white fluffy surface that you see on the top of the cheese is produced primarily by this mold called Penicillium camemberti. So as you might sort of get from this, Penicillium camemberti was originally isolated from camembert cheese. It had been making camembert cheese for long before they ever knew that this was a fungus, and it was named after the cheese that it was originally isolated from. This is a scanning electron micrograph that I took of the surface of a brie-style cheese, and you can see this beautiful kind of paintbrush structure that these molds have. So these are the spores at the tips of these projections that they make on the surface. And what you often get with this type of mold growing on your cheese, you might often notice on this type of cheese that just under the surface, the cheese starts to kind of liquefy, right? So it ages from the outside in. So the longer you let this cheese ripen, the softer it'll get, and it usually is softest just under the rind. And the reason for that is because this microbe is growing on the surface and secreting enzymes into the cheese to eat it. So it really loves to eat casein, the protein. It produces these enzymes called proteases, which chew up the casein so it can eat it for food. And that process of chewing up the casein causes the cheese to liquefy. And it also releases peptides, so small bits of this protein, that can have things like mushroomy flavors. So often when you have this type of cheese with this type of mold growing on it, it has this kind of mushroomy type of flavor. And that, maybe is not so far-fetched, considering mushrooms are another type of fungus. Okay, so they're not that different. Okay, so we have Penicillium camemberti. That's our microbial superstar number 1 here. And then we have its cousin, Penicillium roqueforti. Okay, so who wants to guess where this one was isolated? So from the caves of Roquefort in France. You can see it looks pretty similar. It's a closely related species of fungus. It forms these sort of bluish-green colonies in the lab, and you ate some of it just a little bit ago in this wonderful blue cheese called Point Reyes Blue, also from California, as you can imagine. And the cool thing about this fungus is that, unlike its cousin, it doesn't like to grow on the surface of the cheese. Okay, so to get this mold to grow, so its cousin camemberti likes to grow in lots of oxygen, so it's really happy on the surface of the cheese. However, Penicillium roqueforti is a little afraid of all that oxygen. It likes to grow in low oxygen environments. And so what cheesemakers do is they take the spores of this fungus and they mix it into the milk when they first start making cheese. So the spores are everywhere in the milk. And then they make the cheese. They separate the curds from the whey. You have a wheel of cheese. And once that cheese is made, they take a metal spike and spike the cheese over and over again to let little bits of oxygen into the inside of the wheel of cheese. And so you can see here, this is a really fantastic example. These lines, if you've ever wondered what those were, it's because the cheesemaker took a metal spike, probably by hand. I actually worked for 2 months on a farm. It's incredibly tedious to do this. So the mold is everywhere, but it's only growing where there's little bits of oxygen. So what does it do when it's growing there? We all know the sort of signature flavor of blue cheese. When we eat a blue cheese, we know it's blue cheese. And the reason we know it's blue cheese is because we can actually recognize the molecules that this fungus is producing. So instead of eating protein, it really likes to eat fat in cheese. And it produces a different type of enzyme called lipases, which break down fat. And this releases free fatty acids. So some of these free fatty acids can have these peppery, spicy flavors that we often associate with blue cheeses. So you'll notice sometimes the more blue a blue cheese has, the spicier it is. It's because the more free fatty acids have been released in that cheese. And then it doesn't stop there. It takes those free fatty acids and it uses another set of enzymes called esterases to break these down into volatile compounds, so really small molecules like methyl ketones and thioesters. One of these molecules is called 2-heptanone, and that is the signature flavor of blue cheese. So when we smell blue cheese, we know it's blue cheese, right? It's because we're smelling this 2-heptanone, and the 2-heptanone is a byproduct of the growth of this fungus. So again, we can actually smell this fungus and recognize it as something potentially delicious. Okay. So I'm going to switch over to bacteria now. This particular bacterium is called Brevibacterium linens, and it is thought to be found on this specific types of cheeses called washed rind cheeses. And so during the aging of this cheese, the cheesemakers actually continually wash the surface of the cheese with a salt solution, a brine. And it creates this moist, salty environment that encourages the growth of specific types of bacteria and yeast, usually. This is a picture of this bacterium in the lab. It produces this kind of yellowish-orange pigment. And you can see on these cheeses, they have this pinkish-orange kind of color to them, partly because of bacteria and yeast that produce pigments. You guys ate this a little bit ago. Cowgirl Creamery, which is another wonderful California creamery. Red Hawk is a great example of a washed rind cheese. So these cheeses were originally developed in monasteries in France, it's believed. And these cheeses they are stinky, yes. But they also have a very meaty flavor to them. And part of that is because this bacterium is able to break down protein through a bunch of different enzymes and breaks it all the way down to amino acids. And if you've ever had like a really rich chicken stock or broth, it's because there's a lot of amino acids there. So similar type of thing where you have a lot of free amino acids in your cheese and that gives it a very meaty flavor. So it's thought that the monks who had to undergo periods of dietary restriction and not eating meat during certain times of year, maybe they developed this cheese as sort of a substitute because it had such a meaty, rich flavor. Okay, so however, they don't stop there again. It would be nice maybe if they did, but the bacteria keep going and they produce all of these really small volatile molecules, amines, aldehydes, and sulfur compounds, which can smell fishy, garlicky, and sweaty. So, you may have heard that these cheeses like this smell like feet. In France, they say that feet smell like cheese. It's true, they do. And one of the reasons is because on our feet and in our armpits, we have a cousin of Brevibacterium linens, Brevibacterium epidermidis. And this Brevibacterium, even though it's a slightly different species, does basically the same thing on our body. So we have lots of protein in our body. And this bacteria is able to break it down and produce all these disgusting aromas on us. So that explains why this cheese smells like feet, but somehow we still eat it. Okay. Okay, so the last little microbial superstar that I'll talk about, it relates to Swiss cheese. And so I talk often to cheesemongers who are the professionals who sell cheese, and they say one of the questions they get most often from customers is, where do the holes in Swiss cheese come from? It's kind of a mystery if you've never really thought about it. And unfortunately, it's not these cute little mice. I guess fortunately, but I mean, really cute. It's this adorable little bacterium here. Okay, so this bacterium is called Propionibacterium. And it's kind of interesting because this bacterium will only grow inside the cheese where there's no oxygen. Okay, so it likes this oxygen-free environment. And what it can do is actually use the leftovers of the lactic acid bacteria to grow. So it basically goes in the lactic acid bacterial fridge, takes out the leftovers, and eats it. Okay, so it takes lactic acid. It can ferment this. Even though there's not a lot of energy in this molecule, it does it. And it produces propionic acid, which is that kind of tangy Swiss flavor that you associate with Swiss cheese. A little bit of acetic acid, which is the vinegar, and CO2, which is the gas. Okay, so it's inside the cheese, eating lactic acid and producing gas all around itself. And so what you can imagine is that at the center of each one of these holes, there was a Propionibacterium that started growing and producing a bubble of gas around itself. So that is how the holes get in Swiss cheese. Okay. You guys are going to be so knowledgeable at your next cheese and wine party. Okay. So, let's see. So we have about 15, 20 more minutes, is that? Yeah. So I'm going to spend the rest of the time telling you guys about why I think cheese is interesting to study as a microbiologist. So I don't study cheese just because I think it's delicious, even though that's a perfectly valid reason. I study cheese because it represents an ecosystem. So these microbes that I've told you about are ones that we know a lot about, but these microbes are not growing by themselves on cheese. They're actually growing as parts of communities. And so these are just a few pictures that I've taken in my lab of different species of microbes that were isolated from a cheese. So we basically scrape a cheese, put it on a culture media in our lab, let it grow, and take these beautiful images. So these are molds, these are bacteria. And depending on the type of cheese you start with, you can have a different collection of organisms growing. This one's really nice. This is one of the Penicillium camemberti growing. And these are some, actually, Brevibacterium. Again, beautiful images of these wonderful microbes that live together. This is actually another scanning electron micrograph that I took of the inside of a rind of a cheddar cheese. And all these little, tiny little things here are bacterial cells that are all packed together. And so you can see kind of rod-shaped ones and little round ones. And these are fungal cells of some sort. And they're all really packed in there in what we think of as a community. And if you take a piece of cheese rind and try and count how many species are alive, how many cells are alive in this system, we estimate that for every gram of cheese rind, there's about 10 billion living cells. So it's a very densely packed, very active environment. So as a microbiologist, the field of microbiology has undergone a total transformation over the past 10 or 20 years. And we are learning incredible amounts about the diversity of microorganisms on our planet and how they live in complex communities. And so whether you're interested in the health and ecology of the soil or the ocean, whether you're interested in the human body, who here has heard of the human gut microbiome? It's all the rage these days. In both of these cases, we have incredibly complex communities of microbes living together. So we can have hundreds or thousands of different species living together. which presents an incredibly big challenge for us as microbiologists to understand how these thousands of species actually work together to form communities and have an impact on ecology and health. So what I was interested in doing is thinking, could we actually come up with a simple system to try and dissect how species interact with each other? And so I turned to cheese. So what I'm going to tell you about is sort of the journey my lab has taken over the past few years in trying to establish cheese as a simple model to understand how communities form and how species interact. And so I'll just make sort of a brief argument here for the need for these simple model systems. Much of what we know about biology comes from the study of model organisms. So whether we're thinking about E. coli, which is the organism that I trained to study, whether it's Drosophila, the fruit fly, C. elegans, the nematode, these are all simple organisms that allow us to easily manipulate them and answer very specific questions about how cells and organisms work. However, as I said, in the field of microbiology, we now know that these microbial organisms don't grow by themselves, they grow as part of communities. However, as I mentioned, these communities have incredibly high diversity, so hundreds or thousands of species living together. A potentially even larger challenge than the high diversity is that we don't actually know how to grow many of these organisms in the lab. So the estimates from the soil are that less than 1% of the species of microbes in a gram of soil are actually culturable in the lab. So on top of the fact that there's thousands of species there, we can't even grow them. So how in the world do we study them? And so basically, microbial communities are not good model systems in general. So they're not simple, they're not easy to reproduce, they're not easy to manipulate. So all the things that make model organisms useful are not usually found in microbial communities. So I turned to fermented foods because fermented foods have all the model-like properties that you might want. So they're relatively simple communities that form very reproducibly. We know how to manipulate them, and we know how to grow the organisms in the lab, or at least we think we do. And so whether it's kimchi or sourdough or kombucha or beer or cheese, these are all different examples of microbial ecosystems that could represent good models for studying communities. Okay, so my plan starting out, when I got this position at Harvard, they said, here's money and a lab and do what you want. So we had to map out a strategy there. So our plan was to use cheese as a model. And we wanted to take a survey of the microbial diversity in cheese, because nobody had ever actually done that. We then wanted to show that on top of being able to identify the species present, we could actually bring these organisms into the lab and grow them and start to study them individually. And then we wanted to start to reconstruct these communities in the lab and show we could actually rebuild microbial communities in a controlled way in our lab. So the first project that we worked on was to go out and collect as many cheeses as we could. So we went around, and each of these dots, red dots, represents a cheese that was collected. You can see many here in Europe. This is Vermont here. There's many wonderful artisan cheesemakers in Vermont, a couple in California, and other places in the US. So we sampled cheeses from 10 different countries, 137 different types of cheese. And we had samples from a total of 362 wheels of cheese. So pretty tough field work. And so how do we go from these cheese samples to actually figuring out what lives in them? So we took an approach that has been developed in microbiology and is applied in pretty much every ecosystem on the planet now. So the idea is that DNA sequences act like fingerprints or barcodes. And so we can use these DNA sequences to tell us which species are present in an environment. And one way to think about it is kind of like, barcodes at the supermarket. So you have these two incredibly unhealthy cereals. And how does the checker know which one it is and what to charge you? Well, they scan a barcode, and that gives information about whether this is Lucky Charms or Frosted Flakes. So we do basically the same thing with DNA sequencing. So we sequence the DNA of the organisms that are present. These pieces of DNA act like barcodes. We compare these barcodes to a database, and that database tells us now what was in our cheese. Okay? So we extract the DNA, we sequence it. This is our barcode, basically. It's a DNA sequence, these 4 letters in different combinations. We compare them to giant databases that have, that list. Okay, well, the barcode of this species looks like this, and this species looks different, and that's how we get back a list of names of organisms. And so we did that for our 137 different types of cheese. And I'm not going to go into any detail about this, but this is the rainbow of cheese diversity. Okay, so each of these columns is a different cheese. Each of the colors on this graph is a different species. And so we're able to categorize all of the different bacteria and fungi that live on cheeses from around the world. Okay, so this sort of gave us a starting point of, okay, these are the species that are there. This is who we're looking for that we want to try and study to reconstruct communities. Okay, so what did we actually find on these different types of cheese? So the bloomy rind cheeses, as I told you, are often associated with this mold Penicillium camemberti. It turns out there are other things there as well. And so in addition to Penicillium, we see a lot of this bacterium, this specific type of bacteria, proteobacteria. They seem to like to grow together. Okay, so this is a relatively simple community of organisms. Natural rind cheeses, so these are sort of like aged cheddars. These are aged in caves for a year or 2 or 3, depending on how long the cheese is being produced. This is an example of one of my favorites that's made here in the US, in Vermont. And they have these really rugged looking cheese rinds on them. And they have a completely different type of community than the softer bloomy rind cheeses. So we have different types of fungi present. So there's this really fun sounding fungus Scopulariopsis, which we know pretty much nothing about. This yeast species here, Brevibacterium, which you remember was the stinky feet bacterium. We actually see this on natural rind cheeses, and then a species of Staphylococcus. So in addition to these microbes, one of the things that we also found, and others have described before, we actually have tiny little arthropods on many of these old cheeses. And what these organisms do, they're called cheese mites. They actually, it appears that they graze on the surface of the cheese. They're kind of like cheese cows, like cheese rind cows. So they're basically eating the fungi and bacteria on the surface. And in some cases, these cheese mites are added intentionally to the cheese. So who's ever had mimolette? This is sort of bright orange French cheese that you see. It kind of looks like a pumpkin during Halloween. So the little craters on the surface are actually holes that have been dug out, eaten away by the cheese mites. So they age these cheeses with cheese mites, so the cheese mites can kind of go in and eat away and form this characteristic kind of moonscape on the cheese. And then there's a German cheese that's called Milbenkäse, which is aged in cabinets of cheese mites. And they actually, in the town where they make this, they have a giant sculpture of a cheese mite in their town square. They're very proud of this. So we haven't done much work on this, but my former postdoc took this amazing video. So this is a cheese mite, and this is a mold. This is actually Scopulariopsis. So these are the hyphae that I showed you sort of zoomed in pictures of earlier. And it's eating it like spaghetti.
Why do they have little single hairs shooting out of their bodies?
I have no idea. I know nothing about mites, but they're pretty interesting.
How big are they?
How big are they? I'm trying to think of the actual size.
Tenth of a millimeter.
They're probably like a tenth of a millimeter, something on that order. They look like kind of like dust on the cheese. So you can just barely see them with the naked eye, but if you put them under a microscope, it's quite shocking.
Do you eat the rind with these guys?
You can. Nothing wrong with eating them.
Or protein.
Yeah. So anyway, I could watch this video for a long time. It's on YouTube. It's on our lab website. Feel free to go home and show.
Does it affect the flavor? Does it affect the flavor?
Yeah. So they are thought to impact the flavor in uncharacterized ways. So we don't know exactly what they're doing, but they emit some sort of odor. Yeah.
Waste products.
Probably waste products. Yeah.
Yeah.
Yeah. Okay, so that's my fun aside. Okay, so that was these sort of really rugged old cheeses. So usually you don't see these things until the cheese has been aged for a year or two in a cave. Okay, this wash rind cheese, as I told you, we tend to think of these as being colonized by Brevibacterium. However, when we did our analysis, we actually didn't see any Brevibacterium on these cheeses. Very little around the world. It's sort of an urban legend in cheese making, apparently. So we see certain types of fungi and bacteria. The thing that really stood out to us is these type of bacteria that are normally found in the ocean. So this is not too surprising because this is a very salty environment. And many cheesemakers actually use sea salt to salt their cheeses, so maybe they're coming in from the sea salt. But when we looked at the genomes of these organisms, so the question is, if these cheeses don't have Brevibacterium linens, where is the funk coming from? Okay, so I told you this is the organisms that make stinky cheese. So it turns out that these marine bacteria in their genomes have enzymes that we think could be important in cheese making, including the enzymes that produce this rotten cabbage type of smell. So it's possible that these marine organisms could actually be the major contributor to these stinky cheeses. Still not entirely sure, however, exactly what they're doing. Okay, so this gave us a picture of how cheese diversity looked across many different cheeses from many different countries. And so we wanted to go into a little more detail and think about how these communities actually formed over time. And so to do this, we connected with a cheesemaker in Vermont called Jasper Hill Farm and Cellars. And this is actually the farm that I spent 2 months working on the farm before I started my lab, so I could really learn as much as I could about cheese. It was the hardest work I've ever done in my life. I woke up at 4 every morning, went and made cheese in the cheese house. I worked in the caves. It's incredibly hard labor and tedious work. So
There are caves on that farm as well?
They're caves, yes, I'll show you. Yeah. It's one of a kind place in the United States. So they've blasted into the side of a hill in Vermont and built these giant concrete vaults. So each of these 7 vaults is independently climate controlled, and they age different types of cheese in each of these different vaults. So this is looking into one of these vaults here. So these kind of like airplane hangar type vaults. So what our plan was is to go into these caves and sample cheeses as they were aging, so we can capture what the community development looked like.
Yeah, I guess like the only
Okay, and so we followed this one particular cheese. It's called Bailey Hazen Blue. It's my favorite blue cheese. And you can see just by eye how much it changes over time. So it ages for about 2 or 3 months. And so we wanted to see, is this a reproducible change over time? And is it kind of analogous to something you might see in a forest? So you have this process called an ecological succession, where you have this reproducible type of sets of species that grow early in a new forest and then later species. So we wanted to see if there was the microbial equivalent of this on cheese. So we went in and did all of our DNA sequencing to identify the species at each of these different time points. And it turns out, again, I'm not going to go into much detail here, that we see this reproducible change over time from batch to batch, from wheel to wheel of cheese. And this happens in the bacteria, and it happens in the fungi. So basically the take-home message here is that even though the cheesemakers, in this case, are not adding any of these species to the cheese, these are coming from naturally from the environment, they still have this very reproducible community formation, which is a great property for wanting to study a system. You want it to be reproducible. Yeah?
Are you sampling the rind or
The rind, yeah.
Why the rind?
Is there much going on inside?
Yeah, so the reason why the rind is because there's not that much going on inside the cheese. So inside the cheese you have the lactic acid bacteria, and they're still kind of doing their thing inside the cheese, but it's only, a handful of species. Yeah. I wanted something more challenging. Yeah. So on the rind of cheese you can see there's many different types of bacteria and fungi all growing there. We don't really know much about them. It seemed like a richer system. Yeah.
No matter what I do, when I buy cheese, if I leave it in the refrigerator for more than a couple of weeks, I wrap it in press and seal, I wrap it in an aluminum foil, I spray it with, it still gets mold. What causes? Why?
Because there's already mold on there. There's already mold on there. So the mold really love cheese, basically, and that's one of the one of the things that makes cheese making work.
But what's the best way to keep it from doing that?
I would just cut it off, or you eat it. Yeah. Yeah.
That's what I do.
Okay. So this kind of took us through this first part, and then I'm going to move really quickly through the next two parts. So we characterize the diversity of the system, but this is something that many people in microbiology are able to do quite easily now with the advances in DNA sequencing. So we can just go to any environment and sequence it and know what's there. But we wanted to go a step further and show that we could isolate these different organisms and work with them in the lab. So we looked for, these are the top 14 types of bacteria we find on cheese, and it turns out we can grow all of them individually in the lab. And so I told you in soil, less than 1% of the species are culturable. In cheese, we think we're reaching close to 100% culturability. And these are some pictures of some of these really cute little bacteria, different major groups of microbes here. And we're starting to sequence many of their genomes, which gives us additional insight into what they're doing. And the same is true for fungi, completely culturable. They're even prettier. Many different types of different fungi that we've isolated from cheese we have growing in the lab.
What's the medium that you
So
cultivate?
for most of these, they were just pretty standard lab media. So rich media.
Agar?
Agar. Yes, on agar.
Blood agar?
So similar. So we used either brain heart infusion, which doesn't sound very good, but very rich. Yeah, or a milk-based medium. Yeah. And this, for the fungi, we usually used a sort of yeast extract-based medium. Yeah. And we're starting to sequence some of the fungal genomes as well. Okay, so that was quick. We did that. One of the biggest challenges in most microbial ecosystems we were able to do pretty quickly. But then we wanted to show that we could not only grow them individually, we could recombine these organisms into communities in the lab. And so we developed a method, a very sophisticated method, for taking fresh wheels of cheese. We break it apart after it's frozen, which is not easy. This hammer actually didn't work. We had to basically lift this over our head and drop it on a concrete slab, which our lab neighbors don't really appreciate. We freeze dry the cheese, grind it up into a powder, and basically that's our microbe food. Okay, so our growth media is fresh cheese. And we put it into these plates. These are tiny little wells in a 96 well plate. And we can take from our culture collection now of all these bacteria and fungi that we have in our freezer, mix them together in whatever combination we want, and grow little cheese communities on our 96 well plate that may or may not mimic a variety of fancy cheeses on a cheese plate. Okay, so that's sort of what we're going for. And so we wanted to see if we could actually reproduce cheese community formation. And so we took the top bacteria that you saw in that ecological succession of cheese that we had isolated, and we mixed them back together in our cheese medium. And it turns out we can actually see ecological succession in the lab in our controlled, simplified environment. So you can just see by eye the patterns here are very similar to what I showed you before. Okay, so we went through, we looked at the diversity, we took apart the system, and we were able to reconstruct it in the lab. And that's sort of been the starting point for my lab at UC San Diego. We are now trying to actually dissect the mechanisms for how all these species interact, how it leads to the reproducible community formation, how they fight with each other, how they talk to each other, all the wonderful things that you might want to know about microbes living together in an ecosystem. And so the take-home messages are, cheese is alive, very much so. All of these microbes growing in and on cheese are contributing to the flavor, the smell, the texture of cheese. And in my own research, we're using cheese as a model to try and understand how communities form. And so I barely do any of this work on my own anymore. I have a wonderful team. This is my current team of researchers in the lab, a number of former lab members, collaborators, and funding for our research. So with that, I will thank you for coming.
Do you get your cheese for free?
Yes. Yes. We're actually quite pleased that many cheesemakers wanted to work with us because the aging process, it's based on very traditional practices. Most of the science that has been performed on cheese is really on the lactic acid bacteria and getting that fermentation step to go faster so you can make bigger wheels of cheddar cheese and wrap them in plastic and put them in a fridge for years. So the traditional practices to make these aged cheeses hasn't really been well studied because it's not of large industrial importance. So we still actually, a lot of what happens has been a black box for cheesemakers. And so when something goes wrong, they don't really know how to troubleshoot it, how to fix it. So the hope is that some of the work that we're doing with the interest in basic science will actually inform their cheese-making practices. Yeah.
What is the process that takes place that makes some cheeses strong and some cheeses mild?
Yeah, so it's a combination of time, so how long the cheese is aged, and often moisture, so how much activity, microbial activity you can get depends on how much moisture is in the cheese. So the more activity, the stronger the flavor will be.
And when they age cheese, you talk about it doing like for years.
Yeah.
Until
Quite the right conditions, maybe.
He has the right, really right conditions.
Yeah.
Taking, for example, the many kinds of blue cheese,
Yes.
are the differences in them based more on different biomes or different methods, or are those just two parts of the same thing?
Yeah. So the differences in flavor, in part, are due to the microbiology. In part, are due to the production method, so how the milk and curds are treated, so whether they're heated, whether they're pressed, and in part due to the milk itself, so whether the milk is from, for example, in blue cheese, many, like Roquefort, is made with sheep milk. Sheep milk has its own characteristic flavors. It also has a higher fat content, so those Penicillium roqueforti has more fat to eat, and it produces more of those free fatty acids. So those sheep milk blue cheeses tend to be a lot spicier. So, and also in some cheeses, depending on the time of year they're made, the animals are eating different food. So there's very traditional cheeses made in the Alps that are only made in the summer because that's when the animals are up in the Alps eating grass, and that's when the milk tastes the best, and that's when they make the cheese. Yeah.
I was wondering, why is there toxicity in cheese?
Yeah, so there can be. So there are a couple of foodborne pathogens that are associated with cheese. So Listeria, E. coli, and Staphylococcus aureus. The incidence is quite low because part of the practice of cheese making is the preservation process. But they do happen, so that is a concern. You can imagine that some of these microbial species could be producing toxins of their own. In all the cases that I have read about, for the molds, for example, they don't seem to produce the toxins when they're growing on the cheese, or the species like Penicillium camemberti, which are actually domesticated species, have lost the ability to produce some of the toxins. So actual toxicity seems to be low. Yeah.
Any milk available for consumption today, I think, has been pasteurized. Could you first explain just what pasteurizing is?
Yes.
And then, is there any difference between the cheese made from pasteurized milk from unpasteurized milk?
Right. So pasteurization is a process of trying to inactivate organisms in raw milk. So it's a brief high temperature treatment. So you reduce the amount of organisms present in the raw milk. So traditionally, cheese is made with raw milk. And in the US and in Europe, it's still often made with raw milk. There are many cheeses that are made with pasteurized milk as well, especially large-scale industrial cheeses. The difference in the cheese-making process can be many-fold. So you get some damage to the proteins during pasteurization that can impact the cheese-making process. You can also lose some of the microbial diversity. So I gave you kind of a simple picture of the lactic acid bacteria that are added to cheese, but you have a lot of naturally occurring lactic acid bacteria in milk. And so if you allow those microbes to stay, you'll actually create more flavor in the cheese itself. Trying to think if, does that answer your question? Yeah. So there's an impact on the diversity, the flavor, of the cheese.
Well, on the back of that, I heard that camembert, real camembert in France is defined as only using raw milk.
Yeah.
So that the camembert made in this country, by law, apparently,
Yes.
has to use pasteurized milk, and therefore there has to be a difference.
Yes.
In that cheese.
And there is. So in the United States, you can make cheese with raw milk as long as that cheese is aged for at least 60 days. And the reasoning behind that aging period is somewhat tenuous. There's actually some recent work through dairy researchers as well as the FDA to try and figure out if that actually makes sense. But basically, camembert is usually aged a little over a month, which means by default, you can't make it with raw milk in the U.S. So yeah. So
it's very hard to get the real camembert from France.
Yes.
Here.
Yes, exactly.
In the rind communities, is there anything that's produced that is either toxic or beneficial to the human system? So should you cut the rind or eat it?
Right. Yeah, so I guess I would say that I don't know.
What do you do?
So in some cheeses, I eat the rind. Like in a brie or camembert, the rind is delicious. Most cheeses, I'll kind of taste the rind and see if I like it. Basically, that's my strategy. If I don't like it, I don't eat it. If I like it, I eat it. There's a couple of studies that have happened in France where people are looking at the potential probiotic impact of eating cheese itself. We've done a little bit of work in collaboration with the gut microbiome lab to look at whether these organisms can actually survive the gastrointestinal tract, and some of them can. We don't think you actually get colonized by them, but they can sort of find their way through and still be alive at the other end. And so I think it's a really interesting question for cheese as well as other fermented foods, what exactly the impact is on human health. Yeah.
I presume that beer promotes the growth of bacteria when you're washing the cheese with beer. Mm-hmm. Has there been any work done developing other washes to create different flavors?
Yeah, right. So the washed rind cheeses, I describe them as having this salty brine solution. Often the salt is added directly to beer, so it's a beer brine. But there are some cheeses that are made with cider, a cider brine. I'm trying to think of other...
Any artificial mixes of chemicals?
No, not that I know of, but yeah, you can imagine maybe spiking in certain types of food sources for microbes. Yeah, that's not a bad idea.
Is there any paleontological evidence for how we figured out to make cheese? Has anything ever been found?
Yeah. I have some fascinating hypotheses that I would be happy to discuss. The actual archaeological evidence that we have of the earliest cheese making, I think it's from some remains that were found a couple years ago in Poland, where they found pottery that had little holes in it, which they think were used to drain out the whey, and they could actually extract milk fats from the pottery. So they think it was used in cheese making. And that, I think, was about 9,000 years old. But before that, people had to figure out how did they figure out to use the animal stomach, right? The rennet. That's a mystery to me.
Imagine storing it and just transport it.
Yeah, so the common story is that they were maybe some nomadic peoples. They used animal stomachs as containers. They were transporting their milk. They got wherever they were going, and it was all curdled. So that potentially makes sense. However, I was talking to an anthropologist, and apparently in hunter-gatherer societies, it's considered a delicacy to, if you're able to kill a young animal that has been feeding milk, the insides of their stomachs have curdled milk. Mm-hmm. So basically it's considered a delicacy to bring back the stomach of these young animals and eat the curds out of the stomach. So that to me probably would put that understanding back many, many years beyond storing it. Yeah.
You mentioned that you're studying communities.
Yes.
And that this is kind of the context. And so I'm just wondering if down the road you plan to use this model to study things like the effect of air pollution or other contaminants in our world.
Yeah. So I think one of the bigger goals of my lab is to be able to take this work beyond cheese, right? So, but I don't necessarily know that we need to actually study other systems to do that. I think with many simple model systems, just the ability to generate some basic understanding of the biology, that guides people who are studying more complex systems, right? So we can actually make discoveries in our system that would impact our understanding of more complex systems, and we could potentially test hypotheses generated more complex systems in our simple system. So that's really one of the goals of having a model system, is to be able to use it as a way to generate and test our understanding of the world. Yeah.
Are the cheesemakers interested in, purchasing a biome that's balanced? I'm assuming that back historically they traded starters like the sourdough for the blue cheeses and that sort of thing.
Yeah, so I think there's some sort of modern day interest in that. The farm that I worked with, they actually built a microbiology lab on site because they were so interested in what we were learning. They wanted to try and harness some of that. So there are cheese culture companies that are large industrial companies who produce some of these cultures that are used commonly in cheese, but they usually have a pretty limited kind of portfolio of species. So, yeah.
So I wanted to confirm that you said if you open your refrigerator and your cheese that you now want to eat has mold all over it, you can just cut it off and it's perfectly safe to eat that cheese.
You don't get medical advice.
Yeah. So, okay. So
is this an insurable risk?
Yeah. So what often happens is, especially if you have something like a brie, there's the mold on the surface of the cheese.
It's usually a Parmesan brick.
Oh, okay.
Oh,
yeah. Get a little private here.
Yeah. So I think with something like Parmesan, there's very low moisture, so any chance that things will diffuse much further into the cheese is low. So I would say generally it's fine, but there's probably certain cases where it's not, and I don't know. Okay. Yeah.
Will slime molds grow in cheese? Do you know?
Not that I've ever seen. No.
They don't like that. They like rotten logs and such.
Yeah, but not rotten milk, I guess.
Okay, Sherry, final question. Oh, sorry, well, two.
Oh, yeah.
You just mentioned that the protein casein in the milk is the reason why the milk is so white and not see-through.
Yeah.
But when we have a fat-reduced milk, why is this milk so transparent? Because we reduce the fat and not the protein, right?
Yeah. So part of it's the protein. The other part are large lipid globules that are in the milk. Yes. Yeah. So they're both contributing to the appearance. Yeah.
How did Harvard become enlightened enough to give you?
Really good question. I had some people willing to take some risks on me.
Really?
Yeah.
That's great. That was great.
All right. Well,
Yeah,
thank
thank
you very
you.
much, Rachel. And I'm sure in 5 years she'll come back with a cheese that's called something like Dutton.